For joining us, for those of you who are online. For those of you who are online, I have to say, too bad. You're missing out on some really great food, a free lunch. So that's all right. Thank you for joining us online anyway. I know sometimes that's the only way we can connect. But you're also missing out on one more opportunity. I have four highly valued Beckman Institute stickers. Who hasn't got one that would like one? Well, come on, these are highly valued. I only have two hands up so far. I have one sticker left. Here's the first hand I used. It's so fun to hand these things out. I gotta say, like, I never could have imagined. Well, Bruce is in my spot. So thanks for joining us. It's really a pleasure to have you at our second to last for this semester director's seminars. We are privileged today to have Bruce Damon here, who will be speaking to us about quantitative MRI. Before I introduce Bruce, a couple of housekeeping announcements that I'd like to make. First of all, we're in the middle of a director search for the next Beckman Institute director, and there's three outstanding candidates. Two of them have already been here, and if you missed them, you could see their vision and technical talk online, and I would really encourage people to watch those and provide feedback. Our third candidate, Dr. Alada from Northwestern, will be here on the April 29th and 28th, I believe. And so please, again, participate and provide some feedback on the important decision for the Beckman Institute. Today's seminar speaker is Bruce Damon. He's an adjunct professor of bioengineering here at the University of Illinois, an affiliate faculty member at the Beckman Institute. He's part of our computational imaging group. Bruce, in fact, is a University of Illinois alum. He earned his PhD in molecular and integrative physiology. He went on and did a postdoctoral study at Yale, and then took on a faculty position at Vanderbilt before we were able to recruit him back to the University of Illinois in 2021. At Vanderbilt, Bruce began to establish his research program in using quantitative MRI to look at human physiology and pathophysiology, and especially as it applies to the muscular system. We are, one more thing I wanted to say about Bruce's experience at Vanderbilt. He pointed out to me a very interesting program that he was the director of, a PhD program that's called Chemical and Physical Biology. And I have to say, when I looked at that program, it was very, very inspiring. Bruce described it as, and this is why I think it's so relevant for Beckman, because Bruce described it as what potent power of integrative thinking came out of that program. And really that's why we exist at the Beckman Institute, to integrate and really gain from the power that comes from integrating our cross-discipline activities and experiences. In 2021, during the pandemic, we were actively recruiting, Carl was actively recruiting its director of clinical imaging research, and we were fortunate to be able to find that Bruce was amenable to coming to Champaign and taking up the challenges and opportunities of leading this activity. He, along with Tracy Wozliak, is the co-director of the Carl Advanced Imaging Center, where we are working to establish the 7T MRI, and it's been a real pleasure for me as an observer to watch the two of you stand this effort up and really make an impact for both our community as well as the research that will be able to be happen from that instrument. Today, Bruce is going to discuss quantitative MRI, and he will tell us about, so just sort of if I can plug you into maybe some of the things that you will hear. The use of chemical exchange, I believe, will appear in Bruce's talk. And so, you know, it's something I'm familiar with as a chemist, but I'm really curious to see how that impacts and modulates signals that has meaningful adaptation or understanding for human physiology. And so with that, please join me and welcome Bruce to give our director's seminar.
Well, thanks very much, Jeff, for the kind introduction, and thank you to Jeff and Tracy for the kind invitation. Thank you all for being here, and good afternoon. Being back in town and being here today in particular kind of brought to mind one of the last presentations that I gave at the Beckman Institute. That was in August of 1999 at the annual retreat at the Department of Molecular and Integrated Physiology. So I thought it'd be kind of fun to start by showing you some of the data that I presented on that day and kind of follow the story that's evolved since then. You know, like all PhD education, my education here provided me with those kind of foundational aspects of being a scientist. How we think, you know, our skills, our ethics, and so forth. And then for me also, it was the beginning of the shaping of my scientific philosophy. That it's at the intersection of chemistry, biology, physics, and mathematics that not only can we do quantitative MRI, but also we can do quantitative biology. So I thought a lot of people use the term quantitative MRI, so I thought what I'd do is start by talking about what it means to me, so we have a common definition that we're working from. And then we'll get to the kind of the body of the talk, and I'll wrap up with some concluding comments. So to answer the question of what is quantitative MRI, maybe we should first answer the question, what is MRI? So just to give us, you know, a common starting place, certain atomic nuclei, including protons, have net magnetic properties. MRI is a method for generating, detecting, and spatializing signals that are derived from those net magnetic properties. And that allows us to generate internal images of the body that we can use to study structure or function. How does it work? Well, we won't spend too much time on that, but just to give us a little bit of a common framework. So if we take a system of protons like the body and put it into a very strong magnetic field, the magnetic moments of those protons will interact with each other and their surroundings and reach an equilibrium state. And that equilibrium state is characterized by two things. First of all, the development of a bulk magnetization vector that lies parallel to the magnetic field. And also, those individual magnetic moments are undergoing a sort of orbital motion called precession, much like the wobbles that a spinning gyroscope undergoes under the influence of gravity. To learn something about the system, we need to disrupt the equilibrium, and we do that by introducing energy in the form of a second magnetic field. That will rotate the magnetization vector by any angle that we want, but let's say 90 degrees. And after that, the magnetic moments will continue that orbital or precessional motion. And as they do so, they will induce an electrical current in an antenna that we have placed around the sample. And then what we'll do is we'll use that antenna to observe the system as it returns to equilibrium. And we call that return to equilibrium relaxation. And fundamentally, there are two processes that are involved. First of all, there is an overall return to equilibrium, and we characterize that using an exponential recovery time constant that we call T1. And there's a more rapid loss of signal that is an exponential decay process. And we use a time constant called T2 to characterize how long that takes. And for T1, we're talking, in the overall return to equilibrium, we're talking about 5 to 15 seconds. Signals decay much more rapidly, tens to hundreds of milliseconds at the most. So we're going to be talking about that relaxation process a lot. and the kinds of chemical and physical events that cause it to occur. Tumbling movements of molecules, physical interactions, diffusion, and as Jeff mentioned, chemical exchange.
Bruce, I have a comment.
Yeah.
It's always customary to put the axis going up and down, right, of the magnetic field. But when you slide into an MRI, you're actually, it's horizontal, right?
Yeah, yeah. So thanks for that. Yeah. Okay, so what is quantitative MRI? So we're looking at an image here through the thigh of a patient with a disease called dermatomyositis. We'll talk more about it later. It's an autoimmune inflammatory disease. And you can see that in the vastus lateralis muscle right here. These changes in the image grayscale indicate that the muscle has undergone some necrosis and been infiltrated by fat. And this is kind of a standardly configured clinical radiology image. This is an image of the same patient, in this case, just the right thigh. And here, we can actually apply a specific interpretation to the pixel values. What they represent is the proportion of total MRI signal that is derived from fat as opposed to water. So this is a quantitative image. And to do this, the process looks something like this. We define some biological variables of interest, and we have some signals that we can observe using MRI. What we need to do is define a model that allows us to connect the biology of interest to the MRI signal. And that requires an understanding of the chemical, physical, and biological properties of the tissue. It requires a mathematical model, and our hope is to be able to kind of invert the problem so that we can observe some signals and estimate the tissue properties of interest. So both of those images are matrices and they have numbers in them, but the quantitative image allows us to apply a specific biological meaning to those numbers. So we'll get now into kind of the bulk of the talk, what I've called a relaximetric approach to studying physiology and pathophysiology. So relaximetric just means that we are measuring those relaxation properties that characterize the return of the system to equilibrium. As we go through that, we'll start by talking about the work that I did here, in which I used a model of human exercise to try to understand some of those chemical and physical determinants of contrast in MRI. Then I'll talk about how we use that understanding to develop some biomarkers for muscle disease, and then make a bit of a pivot and talk about how we develop some methods for studying human exercise. The first pivot in my career, I think, at least in my scientific career, came in the fall of 1993 when Beckman had just taken delivery of this whole body imaging system. It was supposed to be for Tesla. When it was being brought up to field the first time, it damaged itself pretty badly, and a lot of graduate students had to make pivots. So I was sitting down with my advisor, Joan Dawson, and the senior staff scientist in the lab, Carl Gregory, and Carl said, well, I found this paper that showed how if you change the breathing gas mixture in a rat, the signal intensity in an MR image changes in a way corresponding to the change in breathing gas mixture. And is there something like that that maybe happens during exercise that you could study using imaging? So I looked into that, and there was a literature that was starting to develop about signal enhancement in MR images of exercise muscles. And so this is what that looks like. This is kind of the seminal paper from a radiologist named Jim Fleckenstein. And so what he had people do a rhythmic hand grip exercise in the magnet, and then he imaged them afterwards. And you can see the signal enhancement here. So physically, what's happening is that we have this signal decay process. And as I say, we characterize the amount of time that it takes for that to occur using an exponential time constant, T2. What's happening to change the contrast in the image is that in the exercise muscles, the signal is decaying more slowly, T2 is increasing. So at the time of the measurement, the exercise muscles just give us more signal than the non-exercise muscles. You've heard me use terms like the signal is decaying more slowly. that it sort of implies a study of the rate of signal decay. And often when we're talking about the basic chemical and physical determinants of signal decay, signal relaxation, it is helpful to speak in terms of rate constants rather than time constants. So for a little while, I'll be talking about rate constants. This is an exponential decay. And so if we were to take these data and put them on a semi-log plot, we get a straight line. Presuming that there is only one chemical species if we had a pure compound, distilled water, something like that. But it turns out if you take a muscle out of a frog and you put it in the magnet, you will get not a single slope here, but rather multiple slopes. And Hazelwood and a bunch of other people showed us that this component here that is decaying most slowly is actually related to water in the interstitial space. This component is related to water in the intercellular space. And if we look really, really carefully at the early part of the curve, we'll find a third component that is actually derived from the hydration or interfacial water that surrounds proteins and other macromolecules. So we have a way to study the organization and exchange of water within tissue. So Jim Fleckenstein was aware of this and did a follow-up experiment in which he studied people who have a disease called McArdle's disease. So this is a genetic myophosphorylase deficiency, which means that people with this disease cannot cleave the terminal glucose from a glycogen molecule and undergo glycolysis with it. So it's very hard for them to exercise. But he convinced them to do that. And what he found was that in the muscles of people with McArdle's disease, there was no post-exercise signaling. So this is about where I came into the story. So this leads to a testable hypothesis that if we inhibit glycolysis in a frog muscle, we would eliminate those exercise-induced changes in the rate of signal decay. So as I say, I did this in frog muscles. For those of you who don't know, frogs are like God's special gift to muscle physiology. So I studied the gastrocnemius muscle here in their calf muscles, and remembers me storing them in the CRISPR. And we put them into one of three different metabolic groups. We treated some with cyanide, which inhibits oxidative phosphorylation. So we expect a lot of lactate accumulation. We treated some with this iodoacetic acid, which inhibits glycolysis, so we expect no lactate accumulation. And then we had a third group that were untreated, and they were really pretty anaerobic. So there was no oxidative phosphorylation for a different reason. But it did control for the paramagnetic effects of cyanide, so that was good. So, in these conditions, if we inhibit oxidative phosphorylation, it's like you have a great job and you got fired, but you got to pay the bills. So you get any job that you can, even if it's a low-paying job, and you need to tap into your savings account. And so we can see the effects of that here. So there's a pretty significant amount of fatigue that happens over the course of 60 muscle contractions. When we inhibit glycolysis, it's more like you had a high-paying job and you got fired, and then you didn't get a job. So you have to go totally to your savings account. So they fatigue really, really rapidly. In terms of the biochemistry, so we did some MR spectroscopy, and in the phosphorus spectrum, you can see some peaks here, phosphoaminoesters, which are mostly glycolytic intermediates, inorganic phosphate, phosphocreatine, that's like the savings bank, and the 3 phosphates of ATP. In the proton spectrum, you can see carnosine, that's the pH buffer, creatine, and then a single peak that is derived both from lactate and lipids. So in the phosphorus spectra following exercise, we see this kind of big accumulation of inorganic phosphate. That's a common finding. It shifts a little bit in terms of its position on the x-axis here. We can use that to measure pH. We see the depletion of phosphocreatine that's going to the savings bank, and there's even a little bit of a decrease in the ATP concentration. Much more dramatic results when we inhibit glycolysis. No savings left, no ATP left. There's a big accumulation of phosphoaminoesters, which means that we're accumulating glycolytic intermediates. And then over in the proton spectrum, the main thing to note here is that the buildup of this peak right here means that we're accumulating lactate. And there's no difference in the peak area down here, so we effectively inhibited glycolysis. And then I guess really the big question for my experiment was, what happens to the rate of signal decay? So first of all, just negative control, take the muscles out, treat them metabolically, stick them in the magnet for an hour. What happens? And the answer is there's no change in the rate of signal decay, so that's a good negative control. When we stimulate them to contract, maximally 60 times, we see these really big changes in the rate of signal decay, as we expect, in the untreated and cyanide-treated muscles. But the big question, and the really critical test of the hypothesis, is what happens when we inhibit glycolysis. And this was the expectation, from the prediction from the hypothesis. But I still remember sitting here in the basement of this building, and it was like 11 at night, and I was analyzing data, and I looked at that result this result for the first time, and it looked like this. And I know that finding is not going to save anybody's life or anything like that, but for me, it was a really cool moment because it was unexpected, and I was aware at that moment that I knew something that nobody else in the world knew, and that was awesome. So the change was lower than when we had inhibited oxidative phosphorylation, but it was still significant. So to a certain degree, I guess you could say that my hypothesis was wrong, but that's okay. I learned a lot from that. So one of the things that I learned is that it wasn't about glycolysis or lactate per se. Rather, it was about changes in intracellular water content that were brought about by the accumulation of osmotically active metabolites. And it was about intracellular acidification that was caused by the accumulation of metabolites with acidic pKa's. It turns out that these two physiological changes in water content and pH, could explain about 70% of the change, of 70% of the variance in the change in the rate of signal relaxation. The experiment it was set up to demonstrate causality with respect to the role of glycolysis. It wasn't really designed to set up to test causality with respect to the role of intracellular water content or pH change. So that was the follow-up experiment. And so we took some sartorius muscles out, put them through some osmotic manipulation to change their intracellular volume, and found this linear relationship between the rate of signal decay and our estimate of the intracellular volume, which is what relaxation theory predicts. So that was kind of cool. We took some other muscles out, manipulated the intracellular pH, and we found a linear relationship between the rate of signal decay and the pH. And I think the linear relationship surprised me a little bit. I was kind of expecting it to look more Henderson-Hasselbalch-y, if that's a word. But it was a linear relationship for sure. The other thing that I noticed, so kind of, I wish I could say I planned this, but sort of by happenstance, I ended up doing some experiments at different magnetic field strengths. And what I found is that these exercise-induced changes in the rate of signal decay varied with the magnetic field strength. So that was a really important clue, as it turns out. And so from all that, a picture began to emerge. So we have some proteins that have functional groups on them. They are in a state of exchange with that interfacial water around the protein. And the interfacial water, in turn, is an exchange with the free water, the water of the cell that is freely diffusible around the cell. And we could come up with more complicated models than this, but this was complicated enough for me, I guess. Importantly, the frequency at which that orbital or precessional motion occurs varies between the protons that are part of these functional groups and the protons that are on water. And back in 1963, Lutz and Myboom showed us that when we have chemical exchange between two different sites with different precessional frequencies, it acts as a mechanism of signal decay. So now the picture was becoming clearer. We had this metabolite accumulation that creates an osmotic force and brings water into the cell, increases the free water content. And that increase in the free water content increases the relative size of this pool, and it kind of pulls up the weighted average of the T2 volume. We have the accumulation of acidic metabolites. That could be lactate, but it could be other glycolytic intermediates as well. So that decreases the pH. And from the literature, we kind of expect that these changes in pH would alter the chemical exchange rate between the functional groups and the interpatient water. Lastly, there was this linear titration of the rate of signal decay on pH. And as I say, I naively thought that was going to look like a Henderson-Hasselbalch relationship. But what the linear relationship probably means that we have a polyprotic acid, so multiple pKa's, or the changes in protonation state of those functional groups could produce some changes in the higher order protein structure as well. And then the last thing to note is that chemical exchange is not the only mechanism of magnetization transfer between proteins and water. There are also three-space interactions between magnetic dipoles that also can occur, but that's probably not the dominant exchange mechanism. So now we kind of move from the transition from my graduate work into my faculty work, where one of the questions we wanted to answer was, through what mechanism does pH affect the rate of signal decay? And we have a technique called magnetization transfer MRI, in which we make the MRI signal intensity explicitly sensitive to the transfer of magnetization through chemical exchange or dipole-dipole interaction between the macromolecular and free water protons. So to do this quantitatively, we'll need to adopt a model that allows us to estimate some parameters that characterize that transfer of magnetization. So we start with this three-pool model, and we can simplify it a little bit by assuming that there's a state of rapid exchange here. And then one of the things that we want to know is, how big is this pool of protons relative to this pool of protons? So we'll use the symbols P sub n and P sub f to characterize the size of those pools, and define a pool size ratio that's going to tell us about the relative protein content of the tissue. The other thing that we want to know is the rate of exchange. Only one of those is an independent rate, so we'll just estimate the rate constant from the macromolecular pool into the free water. So we kind of repeated the experiment with pH. The way that this method works is, the basic idea is if we have a sample of pure water and we do an NMR spectrum on it, we get a single peak. Proteins, on the other hand, give us a very, very broad peak. In a kind of typical MR experiment, we would excite all of those frequencies at once and see what we get. In a magnetization transfer experiment, what we want to do is excite only the water signal or only the protein signal. But because there's exchange between them, the behavior of the overall system is affected, and we can observe that. And there are a couple different approaches to doing this. The details don't really matter too much. If we affect the water pool, we do that with a technique called selective inversion. If we do that in the protein pool, we do that with a technique called off-resonance saturation. This is the one that we started with first, the selective inversion of water. So we did that, kind of made those measurements, estimated some parameters in the repetition of that pH modification experiment in the frog model. And what we found was this. So this is the rate constant for magnetization transfer from the macromolecular into the free water pool. And this is the intracellular pH, and except for this one little outlier down here, we have this linear relationship. And then these are the group means and standard errors down here. And we did this without affecting the pool size ratio. So what that means is that there is an independent effect of pH, both on the rate of signal decay and on the rate of exchange between macromolecules into the free water. So we can add to the picture a little bit. And probably say something about which of these functional groups is the most important partner in this exchange. So it turns out that only amide-water reactions have a pKa in the physiological range, have a difference in resonance frequency that is great enough to be a significant effector of the rate of signal decay, and are base catalysts, which is what we saw in this diagram back here, increasing exchange rate with higher pH. That was about where we got to in terms of kind of like really investigating the chemistry behind that relaxation process. And what I want to do now is kind of make a bit of a pivot to studying human pathophysiology. So a couple things to remember. First of all is that we've seen that if the protein to water ratio changes, that'll change this pool size ratio and the rate of signal decay, or it would increase the time constant for signal decay. We also expect that an increase in the relative volume of the interstitial space would, decrease the rate of signal decay or increase the time constant for signal decay. So those are a couple of important little facts that we can use to think about how could we develop this into a biomarker. I guess we also want to understand something about some typical pathology of muscle disease. Inflammation is typically present as an early component of muscle disease. It's at least partly amenable to treatment using the anti-inflammatory drugs, including steroids, but steroids have some significant effects, side effects. And so, you know, it would be nice to have a way to monitor dosing and so forth. At later stages of muscle disease, as a result of necrosis, fibrosis is often present. So this is kind of an accumulation of extracellular matrix. And it's thought that fibrosis could act as a mechanism for further pathology and could interfere with the delivery of genetic or cellular therapies. And so these are things that we really want to be able to measure and monitor. My colleague Mark Does at Vanderbilt and a number of others have shown that if the extracellular space around cells expands due to edema or something like that, we can observe that by making T2 measurements. So what we're looking at here is kind of the distribution of T2 values within the tissue, with this peak over here representing the expansion of the extracellular space. One thing that we were interested in doing is trying to understand how would inflammation affect potential MRI biomarkers as an isolated feature. So only inflammation is present, not the fibrosis. So we injected some mice in the flank muscle with a compound called lambda-carrageenan right here, and you can see a little signal enhancement right there. This is histology from the affected side and the unaffected side. And you can see there's cellular swelling here. There's an expansion of the interstitial space and kind of the invasion of inflammatory cells there. And then, you know, we made our measurements, measured the T2, the time constant for signal decay, and saw the same kind of behavior, expansion of the extracellular space. And if we model this more simply and just get a single T2 value out, we see a really significant increase. So a very sensitive marker of inflammation. We did a magnetization transfer experiment, see that a decrease in this ratio of macromolecules to free water, which is what you would expect in an inflamed state. And so that and some other measurements we used to characterize the effect of inflammation as a single pathologic feature. Kind of looked into a more complex model of muscle disease, so the MDX mouse, which models is a genetic model of Duchenne muscular dystrophy. And here we see inflammation present, maybe not the best histology in the world, and also fibrosis present in these mice as well. And we looked at the magnetization transfer measurements and saw a decrease, in the pool size ratio in the rate constant for signal decay. Sorry, I paused there. I think I realized that I'm using an old version of the slides. So I'm just going to look at these for a moment. Sorry about this.
No surprise.
We won't know. We'll believe you.
Oh, that's really too bad. You know what I'm going to do? I'm just going to, I'm really sorry.
That's right.
No.
I'm looking at this thinking, how did I change this?
That's when we were trying to do this. I changed ours. Why? 10 minutes before Bruce and I were to present. That's why I was like, I didn't do it this time, but that's normally what happens to it.
Don't blame Tracy.
Fun fact, there's 37 people on Zoom also. So
All right.
I'm monitoring that.
Tell them to queue up their questions.
Yeah, I have, but I won't remind them.
Yeah, the conclusion slides, I think, are a lot. All right, this is better. Sorry about that. Okay, yeah, so doing the magnetization transfer experiment, a decrease in the ratio of macromolecules to free water, and also a decrease in the exchange rate. So with a little bit of animal work behind us, we wanted to think about how do we translate this into human studies. So we put together what we call the kitchen sink protocol, basically every measurement that we can think of. So that included some structural images, diffusion images, magnetization transfer, T1, T2 measurements, and then measurements of fast signal fraction. And these are the ones that I'll tell you about today. So one thing that we would want to do is just make sure that our measurements exert linear behavior. So we took some, made up some gel phantoms of bovine serum albumin at different weight percentages and measured the rate of signal decay and the pool size ratio. And we saw a nice linear behavior over here and not so bad over here. We looked at the reproducibility of all those measurements, did Bland-Altman analysis and the whole bit, and found that for most of those variables, we had reproducibility coefficients of about 5% of the mean or less. So we could feel confident that if there was a difference between patients and controls of more than 5%, we could detect that. So that was true for all variables except that pool size ratio. That turned out to be a very difficult measurement to make in people. So we switched our approach a little bit. I'll just quickly tell you that we ran some simulations and then did some in vivo measurements to make sure that we could get stable estimates of the parameter of interest that didn't seem to be too spatially sensitive. They were fairly stable across the entire thigh in controls and in patients as well. And then we applied this to the study of autoimmune inflammatory myopathy. So this is an axial image through the thigh of a healthy person. This is a patient with a disease called polymyositis, in which muscle proteins serve as the antigenic target. And it produces muscle inflammation, fibrosis, fatty infiltration, atrophy, and so forth. And also patients with a disease called dermatomyositis. This is a B-cell mediated disease in which vascular proteins serve as the antigenic targets. Similar, though less severe muscle phenotype, but because lots of tissues have blood vessels, other organs are affected as well. We made some measurements of the T2 value, and we saw some findings that are consistent with fibrosis. So elevations in our patient groups compared to control. and then a reduction in the pool size ratio, again, the ratio of macromolecules to free water. And then as a downstream consequence of all that inflammation, there is this process of replacing necrotic muscle with fat. And so we saw an increase in the fat content in muscles in those patients as well. So I think one of the things that I learned from this is that, by digging in and understanding the sources of relaxation-based contrast in MRI, we're able to kind of develop new biomarkers based on that knowledge, and also, even for existing biomarkers, interpret the data more quantitatively and more specifically. And also translating small animal MRI methods to human MRI is really, really hard. So the final shift now is to talk a little bit about an exercise physiology study that we did. And this was kind of a fun one. How can we capitalize on what we understand about the determinants of signal decay in muscles to study human physiology? So we know, this is where we started, that exercise in vivo creates the conditions that are necessary to change the rate constant for signal decay, or its time constant. And we saw this in this very first image that we looked at. The specific exercise of interest for us was bicycling, actually. Eventually what we wanted to get to was what is the pattern of muscle activation throughout the entire pedal cycle? What we know about this is that, if we plot out the pedal angle as a function of time over here, and we measure the force on the pedal, it would look something like this over the pedal cycle. And there tends to be an involvement of extensor muscles like the quadriceps in this phase of the pedal cycle. And then when you go to try to pull the pedal back up, the hamstrings, the flexor muscles are more active.
Is it using toe clips?
Yes,
Okay.
so we use toe clips in our study. Yep. If you put somebody on a bike for 15 minutes, and then real quick stick them in the magnet afterwards, you would see the signal enhancement in the muscles that perform the exercise. So the answer to toe clips actually depends on the state of training. So trained cyclists will use toe clips more, and so they would get more activity in their hamstring muscles, whereas untrained cyclists tend to use just their quadriceps to push on the pedal. But what these images tell us about is all the muscles that were active at any point in the last 15 minutes. They don't really have the temporal information about muscle activation as a function of pedal angle. So if this is kind of a typical pattern, so we thought, so what if you changed the loading pattern so that you only loaded the pedal, only provided resistance during this little 30-degree arc up here? For the right leg, then you would see the signal change in the muscles that were active at that time. And in the left leg, you'd see the muscles that were active down here. Then we shift the arc. And if we keep doing that, eventually maybe we could reconstruct the whole pattern of muscle activation. So we took a kind of a standard electromagnetically braked cycloergometer, kind of hacked into the control system with an external control unit and a LabVIEW program that allowed us to kind of trick the system into providing resistance sometimes and not at other times. We couldn't quite we got to like plan F before we made it. So we never got to like 30-degree angular resolution, which was too bad. But what we could do was, first of all, load the pedals from 0 to 230 degrees, which we would expect to require involvement of the extensor and flexor muscles. In another condition, we loaded it from like 90 to 230 degrees, which should be only the flexors. And then the difference between the two creates a virtual condition that should only tell us about extensor muscle. We did some electromyography just to show that this is basically what happened. These are the results. So if you look at the quadriceps from this, what we call the virtual extensor condition, so we get more electromyographic activity in the quadriceps than we do in the hamstrings during that condition. The opposite is true in what should be a flexor condition. Then we made our MR measurements and saw, pretty much the same patterns. So it was pretty good, I thought, for a proof of concept study. And what we wanted to do was figure out how to deal with those angular resolution issues a little bit more. And then, use the muscle activation patterns and force measurements on the pedal to develop a forward dynamic simulation, and eventually use that to improve electrically stimulated pedaling in wheelchair users. So the other thing I learned, though, was that I thought that was really cool, but the NIH reviewers hated it. So that was
What are they?
exactly? No vision. That's the problem. All right. So, we've kind of gone, sorry for the pun, full cycle here. Started by talking about human exercise, kind of dug into some of the chemistry and physics behind some signal changes, and then we've talked about how we can use that understanding to develop MRI biomarkers and study human exercise. So just to conclude a little bit, I've kind of talked about conclusions from each study along the way, and I didn't want to rehash those. So I thought I'd give you some broader. So first, I think that, for MRI in particular, in integrative and quantitative understanding of the chemistry, physics, and biology of the system is essential to being able to perform truly quantitative MRI. And that allows us to develop new approaches to studying physiology and pathophysiology. And it helps us to interpret the data appropriately. And it took a multidisciplinary team to make this work. I have often said, and this was proven to me on Tuesday night again, that I am so fortunate. Like, I want to make these measurements, and I am so fortunate to be able to work with people who are among the world's experts in how to make those measurements well. I think kind of gone are the days when people like my scientific hero, A.V. Hill, would just kind of go into the lab and do things on his own. And he did it all. He was trained as a physicist, so he did the math, he did the engineering, he developed thermocouples, he did the physiology, he did the whole bit. But I don't think one person can do it anymore. Towards the end of his life, A.V. Hill also wrote a book called Trails and Trials in Physiology. And he had a section that was called The Value and Danger of Hypotheses. So, on the one hand, he said, knowledge advances by continual action and reaction between a hypothesis on the one hand and observation, calculation, and experiment on the other. I mean, what a beautiful description of the scientific method. It's elegant. He also said, it is odd how one's brain fails to work properly when pet theories are involved. And I was really lucky, as I say, to have been wrong in this hypothesis. So I was able to kind of appreciate that sort of dynamic tension between the value of a hypothesis in helping you to create an experimental design and make a prediction about your system, but not to become so wedded to it that you feel like you have to prove it. And instead, you just kind of go where the hypothesis test takes you. And I learned that if I had been right, like the only thing that would have happened is that my results would have confirmed my prior understanding. But by being wrong, it forced me to dive deeper into the system and really understand its chemistry, its physics, and its biology. I think at the time, I used to say that I'm studying how the chemical and physical and biological properties of tissues interact to influence contrast in MR images. And I think my model of that probably looks something like this. There's a thing called chemistry, there's a thing called physics, there's a thing called biology, and occasionally they intersect. And we use terms of convenience to describe those points of intersection, like biophysics, physical biology, physical chemistry, chemical physics, chemical biology, and biological chemistry. And we even have, as Jeff pointed out, chemical and physical biology. But I think my view of this has changed over the years. Partly it's been the result of my own work. Partly it's been the result of my involvement in the graduate program, actually. So I've come to see this sort of view as kind of limiting, actually. So what I think now is that chemistry, physics, and biology kind of share a single continuous scientific space, where chemistry, physics, and biology are not sets in a Venn diagram that occasionally intersect with each other. Rather, they are the axes of that space. And, you know, if we could add a fourth dimension to this and make the cube, you know, a tesseract, that fourth dimension would be mathematics and computation. And I think that it's only through that integrative approach that we can have any chance of solving the most important problems of today, you know, let alone in the future. And our education processes, both for our students and for ourselves, need to reflect that. So that's how my scientific philosophy has evolved over the years. And that's why it has been such an honor to speak to you today in this place that is dedicated to that kind of integrated thought. So I will thank all the co-authors on those papers that I told you about, other members of the lab over the years, including some current members, the people who pay the bills, and of course, all of you for your attention, and I'd be happy to take any questions you may have.
Let me start by encouraging people on Zoom to be sure to post questions. And Patty, if you get any.
We have a question.
Go ahead.
All right. Yes. One question is, how did you measure T2 by chance? Was it T2 star?
It was T2. We used a technique called a Carper-cell. I don't know who's asking the question. Oh, okay. Yeah. So it was a Carper-cell Myboom-Gill sequence in the small animal experiments. In the human experiments, this is one of the difficulties. We used so it was a multi-echo, multiple spin echo. I'll just go with that.
Okay, great. Thank you.
Yes. Yeah.
Yeah, have you given any thought about studying muscles that are denervated? You know, like, because early on, as you're probably aware, the EMG takes about a month to show positive results. Sometimes it's very confusing to figure out, you know, exactly what that is.
We haven't done that. I had a couple of colleagues back at Vanderbilt who studied various types of peripheral nerve injury, so including traumatic injury, including transection, and then also diseases of peripheral nerves like Charcot-Marie-Tooth syndrome, for instance. I have to say their primary interest was in looking at the nerve, and then in the regrowth of the nerve in the case of traumatic injury. But they certainly saw, you know, early on a pretty strong muscle effect as well, and actually. Those kinds of fatty infiltration changes that we saw with the autoimmune inflammatory myopathy patients, they would see those probably as in fact, as one of the strongest indicators of the level of nerve injury.
You know how early they showed up?
I couldn't tell you offhand. Tracy.
So thanks so much. This was a wonderful talk. I'm glad you have this image up because I had to lean over to Aaron and say, hey, this is on the left is the image that his daughter thought looked like a slice of ham.
It is the image that my daughter thought looked like a slice of ham.
So this is wonderful. I'm wondering if you could tell us a little bit about the excitement of what you're looking to pursue at doing your muscle work at 7T. What are the challenges and what are the opportunities there?
Yep. Largely that would be a different talk, actually.
Right. Excited.
In a different line of research, we've done some BOLD measurements at 7T, and so BOLD is the contrast mechanism that's used in functional MRI. And so we've done a sort of event-related design, if you will. So we have people do a really short muscle contraction, like 2 seconds long or something like that. And what's kind of interesting about that is that the control system for blood flow doesn't know, if you will, that the person's only going to be contracting for 2 seconds. And that control system uses kind of a mixture of feed-forward control mechanisms and feedback control mechanisms. So in the physiological terminology, feed-forward would be like central command. All it knows is that it's going to exercise. And so it sort of activates these vascular mechanisms that produce a significant blood flow response, actually, even though the metabolic demand of a 2-second muscle contraction is really short. So we can use bold imaging. So bold imaging is sensitive to primarily to the oxyhemoglobin content of the blood. So the balance of oxygen supply and oxygen demand. So in that paradigm, we get this sort of over-response of oxygen supply relative to demand. And so we'll see like a little, for measuring the bold signal time course, we got like a 5% change in bold signal in a healthy person, transiently following that 2-second contraction. In someone with type 2 diabetes or obesity, that response is dramatically lessened, and it's almost a flat response, actually. What's really interesting at 7T is that 7T, as you well know, is so much more sensitive to bold phenomena than lower field strength. So we see this incredible, a larger response and lots of heterogeneity between even healthy people. So we'd really like to be able to understand more about that. What are the specific quantitative contributions of increases in blood flow through the vascular system at the arterial level, what's happening at the microvascular level, and then what's happening metabolically with regards to oxygen demand following the contraction.
All right, I want to, one more question.
Yeah. I don't know if you're so interested in you getting that angular resolution you were looking for, besides, let's say, an iTribute set. But I was just sort of thinking, have you ever considered doing it mechanically? I mean, I'm sure you have your research using those electronic spikes. It seems like it might be fairly simple if you could just have 2 gears and you take out most of the teeth of one of the gears.
Yeah.
You can get the kind of angular resolution you want.
Yeah, part of the reason for that system was we had it, but I could definitely, I could see that as really the way to do it, actually, like those mechanically braked flywheels. I think that would be much easier to control. Just like apply a disc brake to it or pull it away. I think that's probably the way to do it. And I think the way to convince the NIH reviewers is to really show that it's working and that it provides something valuable in the context of wheelchair exercise. So before we go back.
Next question.
I really liked your description about the interaction between biology and physics, so projection into 3D space and vectors and things like that. I have a question. Have you ever been found correlation to lactate?
So what we did was in that experiment was let me go back to that slide actually. Yes. So here in the proton spectrum, so here's like a little bit of a lipid peak, and then here's the lactate here. And so by assuming creatine as an internal standard, so we actually we do the T1 and T2 of lactate. So we were able to actually estimate a concentration change for lactate. And in this condition, this was certainly probably the major osmolate that accumulated. But in the phosphorus spectrum, there were some changes as well. So metabolically, what phosphocreatine does is it transfers one of its phosphates to ATP, and that reaction results in the generation of two osmolytes where you started with one. And so we can look at the change in the inorganic phosphate concentration. And then, from this, between this and the changes in ATP and PCR and the lactate, come up with a total osmolyte change. And that was on the order of, 30% or something like that. I'm kind of making that up. But it was around there in this experiment when we add up the total osmolyte change. And that correlated really well with the T2 changes. So the total osmolyte probably matters more than just the lactate.
Let me stop there and first of all, thank the people online and for those of you in the room. Bruce, I especially appreciated the trace of your career trajectory and for all of those of you who still have years ahead of you, like me, I'm really appreciative of this open-mindedness about how your thinking will evolve over your career. And the other thing is just the stories and sort of the story of being in the basement at 11 o'clock at night and recognizing the many different thoughts that probably went through your mind as this basic idea that you were holding on to as your thesis really had to go and be tossed out the window to come up with some new understanding, and how that understanding ultimately, was really the production of new knowledge. And I think those are incredibly valuable lessons for all of us, no matter what our discipline in science is. And I really appreciate the fact that you took the time to inject those stories into the talk as well. And in addition, a little bit about the future excitement of where things are going. So with that, please thank me in joining Bruce again.